Bringing back the lost, the promise of biological resurrection. Imagine a world where extinct animals roam the earth again, where the majestic woolly mammoth and the curious dodo bird walk among us once more. This is the ambitious goal of biological resurrection technology, also known as de-extinction, a field that combines cutting-edge science with a touch of wonder. Scientists are working tirelessly to bring back species lost to time using advanced genetic engineering and cloning techniques. The prospect of seeing a woolly mammoth or a dodo bird in the flesh is no longer a distant fantasy, but a tangible possibility. This field holds immense potential for restoring lost biodiversity and advancing our understanding of evolution and natural history. By bringing back extinct species, scientists can gain invaluable insights into their biology, behavior, and the ecosystems they once inhabited. This knowledge can significantly aid conservation efforts for endangered species and fragile ecosystems, providing new strategies for preservation. Biological resurrection technology offers a powerful new tool for conservation and scientific discovery, opening doors to possibilities we are only beginning to explore. The journey of de-extinction is just beginning, and the possibilities are vast, promising a future where the past comes alive in ways we never imagined. De-extinction: how science is turning back time. The process of de-extinction relies on advanced genetic engineering, cloning, and cutting-edge biotechnology. It begins with obtaining DNA samples from preserved remains of extinct species, such as bones, teeth, or even mummified tissue. These samples often contain fragmented and degraded DNA, posing a significant challenge for scientists to overcome. Once extracted, the DNA is sequenced and meticulously analyzed to determine the genetic makeup of the extinct species. Scientists use precise gene editing techniques, such as CRISPR, to make necessary modifications to the DNA. The goal is to introduce specific genes from the extinct species into the genome of its closest living relative. This might involve replacing specific genes or inserting entire sections of DNA to recreate the extinct species' genome. Somatic cell nuclear transfer, SCNT, is a promising technique, famously used in cloning Dolly the sheep, and holds great potential for de-extinction. The egg cell, now containing the genetic material of the extinct species, is carefully stimulated to develop into a viable embryo.